Hi, I'm Catherine Chaplin, a senior scientist at the All Wales Therapeutics and Toxicology Centre. This session is a practical guide to reading and interpreting systematic review and meta-analysis. So, why do we need systematic reviews? There is an immense volume of research studies available, with a wide range of methodologies used within these studies. These need to be reviewed and synthesized to support rational interpretations and evidence-based decision-making. A systematic review is a structured research method that collates all available evidence to answer a specific research question. In contrast to traditional literature reviews, there is a rigorous attempt to minimize bias throughout the process of selecting, reviewing, and synthesizing the included research studies. These can be used to help inform treatment recommendations and decisions for patients, which of several treatments is the best for a given condition and a specific patient population. These are some of the key components of a systematic review which you should look out for and be able to identify when reading a systematic review. There should be a well-defined specific research question. Ideally, a protocol including predefined outcomes. This, if it's been published or registered, should be referenced in the paper. These outcomes should be positive and negative, i.e. include safety, adverse events. A clear, comprehensive search strategy, explicit inclusion and exclusion criteria to inform study selection. There should be a table summarizing the patient and study characteristics for included studies and a quality appraisal of these studies, for example, risk of bias. If a meta-analysis has been done, then there will be pooled estimates and an associated forest plot for the outcome. Then there are considerations around the validity and usefulness of the results. For example, clinical relevance of the results and applicability to wider practice. Finally, it is worth checking if any recommendations or evidence have been given a quality rating, e.g. grading of recommendations, assessment, development and evaluation, and whether PRISMA guidelines for transparent reporting have been followed. Broadly speaking, these are some of the things you might want to consider when reading a systematic review. So regarding the research question, how appropriate was the population and how similar was the population across the included studies? This includes things like age, gender, diagnosis, disease stage, treatment stage. Was the search comprehensive? How many and what types of databases were searched? How about grey literature and trial registries? How likely is it that relevant studies may have been missed? Were inclusion and exclusion criteria appropriate? Were studies in certain subgroups of patients excluded? Why might this have been? Was the data extraction process documented? This requires some assumptions to be made. How transparent was this? Did the authors assess the risk of bias for all the included studies? Bias in research is a systematic error or deviation from the truth, occurring at any stage, design, data collection, analysis, publication, and that distorts results. Distorted results can lead to inaccurate conclusions by consistently skewing findings in a particular direction. This is often due to researcher preconceptions, flawed methods, or participant responses, and it impacts validity and reliability. The Risk of Bias 2 tool is commonly used for RCTs. I've included the domains here as a guide, as well as an example of a risk of bias table. The domains have been included to help you identify and understand potential biases in included studies. There are 5 key domains, and some examples of high risk for each of them would be randomization. Patients weren't randomized or an appropriate system for randomization was not used. For deviations from intended interventions, patients and/or personnel were aware of the assigned intervention during the trial, or there were differences in treatment between the two groups other than the intervention being studied. For missing outcome data, proportions of missing outcome data or reasons for missing outcome data differ between the groups. For measurement of the outcome, the measurement of the outcome could have differed between the groups, or outcome assessors were aware of assigned interventions. And for selection of reported results, only reporting outcomes on the basis of the results. Next is understanding the results. So meta-analysis is a statistical technique for combining and weighting the results of individual studies to produce a summary result. Meta-analysis is only appropriate when used as part of a systematic review. Meta-analysis should only be done if patients, interventions, comparisons, outcomes, outcome measures, etc. are sufficiently similar and studies are sufficiently free of bias to produce a clinically meaningful and useful result. Each study must be analysed separately to maintain randomisation. The summary statistics from each study are combined for the meta-analysis. Key components of reporting a meta-analysis are a pooled estimate. For example, this could be as an odds ratio, risk ratio, or mean difference. Confidence intervals, generally 95%. So this indicates the range of values in which we are 95% confident the true value lies. A forest plot is a graph showing each study's effect and confidence interval, as well as the pooled estimate. Heterogeneity is an important consideration, and it is a measure of how much variation there is between study effects. Finally, you should identify which model was used. A fixed effect assumes there is one true underlying effect value and is used when homogeneity is identified. A random effects model assumes the true effect varies between studies and should be used when heterogeneity is identified. This slide shows an example of a forest plot. I'm going to break it down and talk through the key parts. The parts highlighted in blue relate to the individual studies that have been included in the meta-analysis. Each study has a row and includes the raw data, the study estimate, and the confidence intervals. In this example, risk ratios have been used. On the forest plot, the square is the effect estimate and the horizontal line is the confidence interval. At the bottom, we can see the risk ratios. A risk ratio of 1 is equal risk in both groups. If it's greater than 1, then the results favour control, meaning there's a higher risk of the event in the treatment group. A risk ratio of less than 1, there is a lower risk of the event in the treatment group than the control group. The solid line in the middle is the line of no effect. For risk ratios, this had a value of 1. If the confidence interval for an individual study crosses this line of no effect, then the results were not statistically significant. Next, there is information relating to the pooled estimate, the orange parts. Here we can see the total number of events for each of the groups and the pooled estimate. This is represented by a diamond on the forest plot. There is also the test for overall significance between the intervention and the control groups, in which case we can see is significant. So, I mentioned heterogeneity is an important consideration. The test can be found here. In this case, we have an I-squared of 28%. This indicates minor heterogeneity. There is also a chi-squared test, and you can see this is not significant. As there is some heterogeneity, a random effects model has been used. Another indication of heterogeneity is if the confidence intervals of the individual studies do not overlap. So we can see here there is no overlap between these two studies. An I-squared of greater than 50% suggests substantial heterogeneity. Finally, there is the column which shows the weight each study has been given. This indicates the percentage contribution of each study to the meta-analysis result. These should total 100%, and the size of the box on the forest plot reflect the amount of weight given to that study. So we can see in this study example, the risk of cardiac toxicity in a trastuzumab treatment group is 5 times the risk in the comparison group. So now we've been through the forest plot, what questions might we want to ask? How many studies were included in the meta-analysis? How big were the included studies? What direction were the treatment effects for each study? Were they consistent or inconsistent? What is the direction of the pooled effect? Was a fixed or random model used? If there was evidence of heterogeneity, were there key differences in study design or inclusion criteria? How was the outcome defined or measured, and did this vary across studies? If heterogeneity is found, this can be investigated to see if it can be explained using subgroup analysis for discrete characteristics or meta-regression for non-discrete or continuous characteristics. This is to see if the characteristics of studies may be associated with treatment effect. In a subgroup analysis, trials are grouped according to a patient or study characteristic, could be age, dose, and trials within each group are meta-analysed. In some of the trials comparing trastuzumab-containing regimes with regimes not containing trastuzumab, the trastuzumab was administered after chemotherapy. In other trials, trastuzumab and chemotherapy were given concurrently. Subgroup analysis separates these trials. The overall effect is also shown at the bottom. The outcome for this analysis was overall survival. The forest plot contains the same information as before. Here we can see the test of the overall effect was not significant for the sequential trials. The test for concurrent trials was significant, with the hazard ratio favouring trastuzumab-containing regimes. The overall effect is also significant, but the test for differences between the subgroups was not significant. Reporting biases arise when the dissemination of research findings is influenced by the nature and direction of the results. Statistically significant positive results are more likely to be published in English, published more than once, published in high-impact journals, and cited by others. Data that would lead to negative results may be filtered, manipulated, or presented in such a way that they become positive. Reporting biases can have three types of consequences for a meta-analysis. The systematic review may fail to locate an eligible study because all information about it is suppressed or hard to find. A located study may not provide usable data for the outcome of interest because the study authors did not consider the result sufficiently interesting. A located study may provide biased results for some outcomes, for example, by presenting the results with the smallest p-value or the largest effect estimate after trying several analysis methods. These biases, called funnel plot asymmetry, if statistically significant results suggesting a beneficial effect are more likely to be published than non-significant results. An example of a symmetrical funnel plot is shown at the top of this slide. The outer dashed lines indicate the triangular region within which 95% of studies are expected to lie in the absence of biases and heterogeneity. The solid line corresponds to the no intervention line. Larger studies tend to provide smaller effect estimates, i.e. they're closer to the solid line, smaller standard errors, i.e. more precise effect estimates, and they cluster together at the top of the funnel plot. Smaller studies tend to provide less precise effect estimates, larger standard errors, and often larger effect sizes, i.e. odds ratios are much further from 1, and they scatter widely at the bottom of the graph. Smaller studies may provide more extreme results to larger studies because they may be conducted at an earlier phase of clinical trial development or be lower quality due to less funding, etc. Asymmetry may be exaggerated if there's a further tendency for smaller studies to be prone to selective suppression of results than larger studies. This is assumed to be the case for randomized control trials. For instance, it's probably more difficult to make a large study disappear without trace, while a small study can easily be lost in a drawer somewhere. The same may apply to specific outcomes. For example, it is very difficult not to report on mortality if this is an outcome of a large study. Non-reporting bias may occur if evidence from small studies in the wrong direction are not reported. In this plot, we can see there is a suggestion of missing studies in the middle and right of the plot, broadly in the white area of non-significance. This shows that publication bias is plausible. So, we've looked at the methodology and the results. Now we can think about overall validity and usefulness of the review and the results. Clinical relevance. Do these findings matter in practice? What does a risk ratio of 0.5 mean? It's important to understand the baseline. For example, a risk ratio of 0.5 could represent a very significant clinically important reduction in events from 80% to 40%. It could also mean a much smaller and less clinically significant change from 0.4% to 0.2%. Also think about applicability. It may be a very well done systematic review and meta-analysis, but how similar is it to your patients in your context? How generalizable is it? And how might patient values affect the harm and benefit balance? So that's a whistle-stop tour of systematic reviews and meta-analyses. However, most of the examples have been for systematic reviews of RCTs. However, it's worth highlighting the principles and considerations applied to other types of studies and contexts as well. For example, observational studies, diagnostic, prevalence studies. Some challenges listed here, including defining a study, defining a result, risk of bias in individual study results, searching for studies, publication bias, heterogeneity, getting consistent types of results from each study, and dealing with adjustments for different covariates. That being said, these can be very useful, especially when RCT data is not available, is incomplete, or infeasible. Finally, I'm going to briefly show a network meta-analysis. The conventional meta-analysis approach is useful, but it's limited in that it can only compare 2 interventions at a time, and only those evaluated directly in head-to-head trials. For example, RCTs comparing A and C, or a separate meta-analysis looking at RCTs comparing B and C. Network meta-analysis has many advantages over regular meta-analysis when comparing more than 2 interventions. You can compare all interventions in a single analysis, and you can produce treatment effects for all possible treatment pairings. Specifically, it produces results even when not all pairs of interventions have been directly compared in trials. The indirect evidence refers to evidence obtained through one or more common comparator. So if there are no RCTs that directly evaluate A versus B, however, we have direct comparisons for A versus C and direct comparisons for B versus C. These can then be used to calculate the effects of A versus B. These models can be very large and complex. So this is an example of a network diagram for glaucoma treatments. The lines show all the direct comparisons. The size of the nodes or circles represent how many patients received each of the treatments. The width of the lines represent the number of trials that have compared those 2 treatments. Any treatments without a line connecting them can be compared in a network meta-analysis. So what are other factors related to a network meta-analysis? So transitivity. All the treatments included in the network meta-analysis must be suitable for all patients. i.e., imagine it was a hypothetical RCT. Could patients be randomized to any of the arms? For example, a network meta-analysis should not include treatments for HER2-positive breast cancer and HER2-negative breast cancer treatments, as HER2-positive patients could not be given HER2-negative treatments. A network meta-analysis allows for ranking of interventions. These may be displayed in a table or on a plot. These will show each intervention's probability of being the best. This is commonly reported using surface under the cumulative ranking curves, or SUCRA for short. This value represents the probability a treatment has of being among the best options. A SUCRA value of 100% indicates the treatment is certain to be the most effective in the network, while a value of 0% indicates it's certain to be the least effective. The larger the SUCRA value, the better the rank of an intervention in the network. In terms of interpretation, results need to be interpreted with caution for a number of reasons. The presence of heterogeneity, inconsistency in the network, poor underlying quality of studies, and only a small amount of data being available. Check whether PRISMA guidelines for network meta-analysis have been followed. Rankings, like SUCRA values, while appealing to inform clinical decisions, may be misleading. High-ranking treatments may still only have modest or insignificant clinical effects, and therefore should be interpreted in the context of the treatment effects. The SUCRA message can provide a distinct rank for each treatment, but within-class treatment effect differences may be small and potentially not clinically meaningful, indicating that factors other than effectiveness, e.g. costs, side effects, patient preferences, may be more important in treatment selection. The conclusions drawn from network meta-analysis should depend on the outcomes examined, so they should consider both effectiveness and safety outcomes, and whether outcomes evaluated were surrogate outcomes or clinical or patient-important outcomes. In conclusion, to make the results of a systematic review, meta-analysis and meta-analysis useful to researchers, clinicians, decision-makers, and patients, it is important to ensure that the entire process is transparent so that the strengths and weaknesses can be properly evaluated. Some indicators of strong evidence may be consistent results from high-quality, low risk of bias studies, especially RCTs for interventions. Indicators of weak evidence may be inconsistent results or high risk of bias in included studies or small non-randomized studies. I hope this session can help you move beyond simply reading the abstract to critically understanding how much you can trust the review's conclusions for making informed decisions. Diolch. Thank you.